Happy New Year. It's been so cold over the entire country this week. Here on Vashon Island where I live, wind chill got well below zero, and some of the southern states had wind chill of like -40 and -60 degrees Fahrenheit. That's ridiculous. When it gets this cold, my old house is miserable. The poor insulation here just lets the cold right through, and I have to bundle up and keep moving until the weather changes. Several parts of the country are without power too. Cold and no electricity makes life dangerous and unbearable. The parts of my island that have no power have put their freezer foods outside in the icy cold to stay fresh. Some folks are grilling their defrosting expensive meat and seafood so they can enjoy eating them before losing them in case the weather warms before the electricity comes back. Some folks are even leaving town to stay with relatives. And believe it or not, this is the theme of today's Shaping Fire episode. Just like humans get miserable in crappy weather, so does the microbe life in your cannabis soil. Too much water, too little water. Not enough sunshine, too much sunshine. Too little carbon, too much carbon. Too hot, too cold. Frozen. Yikes. I imagine the microbes also putting all their little foods out in the front yard and having the neighborhood over for a big freezing barbecue. Yeah, that's right. Get ready for a lot of anthropomorphizing of microbes today, because microbes are people too. If you want to learn about cannabis health, cultivation, and technique efficiently and with good cheer, I encourage you to subscribe to our newsletter. We'll send you new podcast episodes as they come out, delivered right to your inbox, along with commentary on a couple of the most important news items from the week and videos too. Don't rely on social media to let you know when a new episode is published. Sign up for the updates to make sure you don't miss an episode. You are listening to Shaping Fire, and I'm your host, Shango Los. Welcome to episode 112. My guest today is Michael DiLegge. Michael DiLegge earned his master's from Colorado State University's horticultural department in 2020, where he researched different facets of plant and microbe interactions, including nematode biocontrol methods, microbial legacies resulting from replanting fields, and even the caterpillar gut microbiome. Now serving as the director of microbiology at Impello Biosciences, his research surrounds the overlap between biological and biochemical agricultural inputs targeted towards both crop and soil health properties. Cannabis sativa is one of their model organisms for agriculture research, and one of Michael's projects is to examine the functional bacterial species recruited by cannabis sativa in the rhizosphere. During the first set today, we will discuss biostimulants and plant growth regulators and how to approach them as regenerative cannabis cultivators. In the second set, we will discuss the care and tending of microlife in the rhizosphere. And during the third set, we will choose companion plants for soil diversity and finish the episode with an indictment of the 24-hour light strategy. Welcome to Shaping Fire, Michael.
Hi, Shango. Thank you so much for having me. I'm very grateful to be here.
Awesome. Really glad that you made time for us. So let's get right into it. You know, cannabis farmers are always looking for like a leg up, something winning to add to their recipe. And a lot of us look for different things that could be determined to be biostimulants. And then a subsection of those people are people mostly who follow this show who are regeneratively minded farmers. So they're looking for natural biostimulants versus synthetic ones. Let's start out the show by talking about what biostimulants are generally, and then we'll start to tease apart the synthetic ones from natural ones. So that's my first question for you. What are biostimulants, and what are the kinds of attributes that we look for them to impart on our cannabis plants?
Absolutely, yeah. So, I like to think of biostimulants as a really large umbrella term with several different kind of facets of products or inputs that people can use or they can take advantage of that might be in a natural system. But I guess in essence, they're very much performance enhancers for plant growth and metabolism, is how I kind of define biostimulants.
So when we say performance, are we talking about specifically like the gathering of nutrition, the processing of nutrition, improved photosynthesis, essentially all of the plant's biological systems get kind of like tweaked up. The governor is turned up on all of them, so they're running either more efficiently or faster. That's the idea?
Yeah, I think that's a really good way to put it. And, different parts of biostimulants will upregulate different parts of the plant metabolism or growth rates and things like that. But yeah, I think primarily I look at these as, biological products, things like microorganisms and biochemicals, things that are usually plant derived or discovered in nature and then used to upregulate the plant metabolism, whether that's with hormones or amino acids, fulvic acids, things like that.
I like this term upregulate. I've heard it before, but I don't use it regularly. And I like it both for the biology conversation, but it also sounds like a great euphemism for getting high. Like, oh man, I need to go upregulate.
That's awesome. I like that a lot.
So let's tease apart now the synthetic and the natural biostimulants, because we're mostly going to be talking about natural biostimulants today. So when I discuss natural biostimulants with other regenerative growers, we're usually talking about, this or that plant that we are going to, do a tea of, or an extract of, or a top dress of, something like that, because we want some naturally occurring biochemical that's in the plant. We want that to seep into our biosphere. And then when at the conventions, when I talk with the synthetic biostimulant sales guys, their products have always got these like multi-syllabic names that sound very scientific, and sometimes they've also been named at some marketing name, and so they have got a real different vibe to them. Perhaps the natural ones and the synthetic ones are both trying to do the same things, but I'm guessing they have got different attributes that you can share with us.
Yeah, absolutely. So I would think like a lot of times when I think about synthetic biostimulants, whether that's something that is discovered through a natural process and then one compound is isolated, it really seems a lot to me like the pharmaceutical industry. We find one active ingredient, we purify it, it shows in a lab and on the bench that this is effective at whatever we're trying to alleviate. And then all of the, medicine is built off of that compound. And that's kind of how I look at like the synthetic aspect of that, where potentially there's a chance of missing some other minor chemicals or, metabolites that might be from a natural product extract that would be missed when things are just synthesized in a lab versus extracted through a natural process, like making a compost tea or breaking down other forms of organic inputs like fish waste or even corn waste, things like that.
Hearing you describe it, it actually reminds me a lot of how we talk about the entourage effect and the difference between isolates and whole plant. It sounds a lot like the synthetic biostimulants are often like isolates. They are an isolated biochemical that we're going to apply to the plant one way or another, versus the compost tea kind of flavor. That's more like taking a whole plant medicine, because we're getting all these random plant constituents that we may not have even researched yet, but we know that as a symphony, they interact really well with the human body. What do you think about that?
Yeah, I would agree with that. I think it's really similar to how people talk about the entourage effect of cannabis and terpenoids and things like that. The same thing goes for whether these compounds, whether a synthetic or natural biostimulant is directly influencing the plant. There's a good chance it is. It's also really helping out like the soil microbiome, most likely. Things like the upregulation of the plant metabolism can be a factor of an increased rate of soil respiration or soil organic matter or just general beneficial soil ecological components. Things like amino acids, the things are one of the smaller terms underneath the larger biostimulant umbrella, can become building blocks for protein synthesis in both plants and microbes. So by providing that source that might be deficient in the soil or outcompeted by other microbes, if a grower can add that in there, whether that's helping the plant directly or the soil ecology, the whole system is benefited, at least from what we've seen.
Mm-hmm, mm-hmm. So I do recognize that I've got a certain amount of prejudice against synthetic biostimulants simply because they are synthetic. And I know that that is often unfair of me just to discount things because they're synthetic. Outside of the fact that just on its face, I prefer organic gardening and organic food and organic seeds because I like to, have my garden be as close to nature as possible. Is there really any a priori reason to discount or not include synthetic biostimulants? And what I'm going to compare it to is, we often talk about, bottled salt fertilizers on this show about how they kind of force feed nutrition to cannabis plants. And by doing that, you might get a robust plant, but you're also getting a plant that is out of balance and over the long term is more susceptible to plants and can have all sorts of other problems because you're forcing it, right? Now, are there any mechanisms like that with synthetic biostimulants that are a reason to reject them because of how they function instead of just me being prejudiced because they are not organic?
No, I understand where you're coming from with the synthetic prejudice, and I kind of adapt similar principles like in my own gardening and things like that. I guess I want to ask you a question about what might be considered, synthetic, to take a step back here. I'm familiar with a process for an amino acid product that is taking waste from the fishing industry, so lots of shrimp shells and crab shells and things like that, and then they're put into a reactor. A couple enzymes are added to break down the fish waste or the crustacean waste, and then the end product is both amino acids and chitin and other things like that. So that is, synthetically, the breakdown process is synthetically increased with these enzymes in the reaction. But if you were to kind of slowly compost, shrimp and crab shells and things like that in your soil, you might have a similar endpoint product or, beneficial effect of the soil system. So I guess, how do you feel about that kind of process? Is that, would you call that more synthetic, kind of towing the line between the two of them, or just is that purely synthetic because, lab equipment and bioreactors are involved?
Yeah, it's an interesting question to kind of tease the definition. Definitely, from the top, I'll say the word synthetic is a messy term because it's kind of like it's a bucket of ideas that are different to pretty much everybody who hears the word, and it is best applied on a case-by-case basis. Based on what you are, based on this little example, though, it sounds like that is a non...
It's tricky, right?
It is tricky. It sounds like it's a natural process, right? It sounds like you're just processing by adding other natural processes. So you've got naturally occurring product A, and you're adding naturally occurring products B and C, and you put them together, and it causes the breakdown to happen into a usable nutrition faster. If we were to go long on this and make it the brunt of the show, I would probably have to ask a bunch of questions about what are the natures of the inputs and where that line might be drawn between adding inputs that either inhibit natural reactions or create non-natural reactions to get whatever you're trying to produce. But that's a slippery slope right there, of course, right?
Absolutely, yeah. I think the processes would theoretically occur in nature, just a little bit longer time for those enzymes to unlock some of the polymers, like things in the exoskeletons of crustaceans. We are synthetically, or in this type of process development, somebody would be synthetically upregulating or increasing the rate of decomp to make these things in a more plant usable form. That way the plants and the soil microbes have to expend less energy on breaking down things, and they can just be slurped up and used efficiently. I think it kind of toes the line between synthetic and natural. But one thing that is kind of cool in processes such as these, I know there's similar ones for corn and soy based biostimulants. They do put an end use product to things that might be considered waste material prior to these types of reactions.
And I guess we just want to make sure that waste material is handled in a responsible way if we're going to produce it at all.
Absolutely. And, when it is not waste material, when it's, host material for a crustacean farmer or a corn or soy farmer, we want to make sure that those plants or animals are being treated, and, the management practices of cultivating those are also not going to influence the inputs that we want to feed our plants later.
Mm-hmm. So let's talk a little bit about some of the natural biostimulants that come up the most often in cannabis cultivation. I'm thinking humic and fulvic acids, seaweed extracts, different types of microbial inoculants, and then amino acids, which you've already mentioned. Are these different types of natural biostimulants working on different biological systems of the cannabis plant, or are they all essentially acting on the same system but in different ways?
Yeah, they would definitely act on the same system as a whole, the plant, the soil, the microbes, and whatnot. But every part, everything you just mentioned, has a slightly different effect on the biome. From what I understand, humics and fulvics can be great sources of organic matter for both the plant and the soil microbes to use as inputs, whereas things like seaweed extracts often contain plant hormones like cytokinin content and I think some phosphorus or potassium as well. So we get a little bit of plant nutrition from there, as well as some plant hormones applied outside of the plant body rather than being produced within it. Microbial inoculants, I think these guys really depends on your soil and the history of that system or your indoor cultivation system. But those will primarily, if they're administered in a drench, be benefiting the root systems of the plants as well as the neighboring microbes that might already be there. And then we have, foliar spray microbial inoculants too that help outcompete things that might be negative to plant performances like molds and stuff like that. So yeah, it really is a whole system of effects. Different building blocks from the biostimulant world will help different areas of the plant, as you mentioned. And it's a fun big umbrella term because there's so much going on underneath the umbrella that is biostimulants.
Mm-hmm. It also sounds like it's a very fine line between biostimulants and nutrition, because some of the positive attributes that you mentioned from seaweed extracts are different, nutritive substances. Is there a scientific line between nutrition and biostimulants, or do they kind of like seeg into each other a lot of times?
I think the line is maybe dotted as opposed to less of a gradient, less of a firm line, where a lot of biostimulant compounds can have some form of NPK or even micronutrients in there. But then when we look at plant nutrition, those are products with NPK is almost the biggest number on the label besides the product name for the most part. So that's kind of how I see the separation between, nutritional inputs versus biostimulant based inputs. But it is kind of a gray area or a gradient where you have some trickling effects of the amino acid type biostimulants having some form of NP or K in them as well.
Yeah, using that definition, biostimulants becomes really, really close to like trace nutrients and trace minerals. It's like, if you're not talking about NPK and you're just talking about everything else, you're talking about biostimulants. Including things that we don't get in kind of targeted generic fertilizers that we add because we want to round out the rhizosphere with essential microbes.
Yeah, absolutely. And one other thing to mention too is whether or not. Another reason, I guess, that the line between nutrition and biostimulation is maybe a little bit more blurry at this point in science is because things like certain bacteria have enzymes to unlock unavailable forms of phosphorus, such as calcium phosphate, so unavailable for plant uptake. So there's no nutrition in that type of bacterial product, but it can help unlock nutrition that might already be in your soil. Things like mycorrhizal products, too, I think, from what I've seen, also sometimes have a phosphorus content as well. So, somebody might apply a mycorrhizal product for the first time and see a really great, flowering boost in their cannabis plants. And it's hard to attribute that to the mycorrhizae being present or the increased amount of phosphorus being also available for the plant, or probably both.
Yeah, I wish that when we tried a whole bunch of new things at the same time and we get good results, we could just straight up ask the plant, which one caused it, my friend?
That would be amazing, and yeah, I want to talk to the soil microbiome as well. I'm looking forward to those developments, maybe with AI.
There we go. So before we move on from biostimulants, let's bring this conversation and make it like real rubber hits the road. So for the cultivators who are listening, who are like, okay, I understand the difference between the synthetic and the biostimulants as much as we can get to definitionally. I like the idea of offering natural biostimulants to my plants. What are some that you recommend from the natural world that regenerative farmers can use at home that will have, reliably good effects? Some of the stuff is so obscure and its impact is so small that you can have a debate about whether some of these did anything at all in particular. But what are a couple like tried and true naturally occurring biostimulants that that our listeners can explore for pretty assured benefit?
Yeah, I mean, I think a lot of it really depends on where your plants are going to be going. If it's going into a hydroponic or controlled environmental system versus a soil system, plays a role. But I am biased to the soil microbes and the root adhering microbes or the plant growth promoting microbes.
So are we here at Shaping Fire. So
yeah, absolutely. From what I've seen in both research and at home cultivation, the sooner microbes get introduced to a plant root, I like to think about it as a really good protective and enhancing form. One of my favorite analogies here is think of that radicle, that firstly emerged plant root from your seed or your clone, being like an empty seat at a table with, let's say 100 seats. The plant is going to be exuding root exudates all over that table and through the roots. And those are basically like adding different types of food sources to the table here. The sooner that those seats at the table can be colonized with plant benefiting or soil ecology benefiting, or even just free living microorganisms, maybe not colonized, but present and taking up space and competing for resources, the more likely the cultivator would be, not seeing things like plant disease. If the seeds can be filled with things like plant benefiting microorganisms, whether they're from a compost system, living soil, indigenous microorganism cultivating, or even a biological inoculant you can buy on a shelf, I think that is probably the best way to really prime young or new plants for a successful harvest cycle. Filling those seats up with the table, allowing those resources to be used by things that are maybe symbiotic or mutualist with the plants to, the benefit is a feedback loop. Whereas a pathogen might only, consume a root exudate until it can get in high enough numbers to penetrate the plant cell and then become pathogenic. The less seats at that table that are for things like Fusarium or Phytophthora or Pythium, the less likely, that plant is going to have a negative effect.
I really like that seats at a table example. That I can really envision that. And so your first answer is to add a bunch of indigenous microbes so that the seats of the table can get filled with probiotic folks before anything pathogenic shows up. And and there's a range of ways that we talk about on this show to add microbes, and you're like, just add them right away as soon as the plant starts. For the second one, is there something that might be more nutrition based instead of like having good neighbors based, like microbes, that you would also recommend as a naturally occurring biostimulant?
Yeah, so I think my bias is really going to show with this answer. But, I talked about the probiotics, right? I also think that prebiotics are just as important, especially in soilless systems or near sterile systems, right? Hydroponics and things like that. Plants evolve roots with their relationships with microbes. Dr. White and all of his research surrounding rhizophagy has really been a beneficial disruption in agricultural research. Yeah. And I think that a lot of growers have tried really hard to maintain a sterile system, which, your plants don't want sterile systems. They're often, exuding cakes and cookies, sugars and carbon into the surrounding soils to try to get microbes to colonize there. So a long way to answer your question, I think like prebiotics, things like organic materials, humic fulvic acids, we know what those can do to benefit the plant, and how they are benefiting the soil microbiome and how that's benefiting the plant is still being kind of fleshed out. But I do think that your plants are going to benefit from these compounds that you're applying, whether or not they're for the plant or the bacteria. And usually the application rates from things, whether you're whipping up a compost tea or doing like a plant extract or purchasing something, there's enough to go around for both the plants and the microbes.
Fantastic. I love that when we keep on coming back to, grab plants that are already in your yard and let them decay in water, spin that for 36 hours and then pour that back on your plants. I love that cycle, bringing back, what's already in your yard, the plants, the grass clippings, the forest duff, whatever it is, incubate it for a little bit to make those microbes super abundant and then just pour it back on your plants.
Yes, absolutely. Yeah. I mean, that, and it's always, a fun term is like kind of bioprospecting, being out in soil, in the woods, in the forest, or anywhere that, it's okay to kind of, I'm going to use the word sample from. I know you guys have talked about indigenous microorganisms here, but yeah, like the ability to kind of collect some microbial diversity from maybe a forest or somebody with a better historical soil health practice than maybe the plot you inherited is also a great way to, add that microbial diversity. If we have these like notions of plant microbe symbioses in science where, we want to look at a really happy cannabis plant and then collect some soil microbes from its roots and then take that home and apply it to our cannabis plants so they do well too. But there's a lot of functional redundancy in the rhizosphere microbiome world, whether that's a palm tree or a cannabis plant or some grass. So you might even be able to get some type of beneficial microbial function from sampling something like grass and applying it to your home grow, or making a compost tea out of it or a soil slurry.
Right on. So, dear listener, if you're very interested in moving these microbes around, sit tight, because we're going to be coming back to this topic again with set 2. So, Michael, I want to move us on past biostimulants now to what I consider one of their cousins that I know less about, which is plant growth regulators. And, I first got turned on to plant growth regulators kind of as a slur, as a diss, when I would come across like really rock hard indoor rock wool buds, and that I would squeeze them, and as a lifelong outdoor grower, when you squeeze my flowers, there's some give to them, whereas these were like literally rocks, and I was all like, what kind of cannabis is this, right? And people are like, because I tend to hang out with, organic-y farmers, they'd say something like, it's those PGRs that they use, and I'm like, I don't know what those are, but I certainly don't want any part of that. And over time, I realized it's true that synthetic PGRs and what they have a tendency to do to the plant are not something that I'm normally going for. But then I started to realize, while studying the soil food web, that there are actually naturally occurring biochemicals in plants that that we can wildcraft that also are plant growth regulators, so that maybe you can get harder flowers that have got a market attraction and an aesthetic attraction in ways that don't also injure the plant and make it, need to be grown in a sterile environment instead of the yard. So I'm going to ask you, number one, would you explain for us what plant growth regulators are and where we may find them in the natural world?
Yeah. So the way I kind of understand plant growth regulators would be just kind of hormones, the plant hormones, things like auxins, gibberellins, cytokinins, and natural sources of those things. Like seaweed is a really common input used for cytokinin, PGRs. I think, like we talked about a little bit earlier, when you do the wildcrafting and you do the natural process and take your plant inputs or get materials and do the extraction yourself, you are likely getting things that might be missed, as opposed to when an auxin is purified in a lab and dried out into a powder and then, mailed to you in a tube to dip your plant roots in before rooting them. The way I kind of see them would just be plant hormones, whether they're natural or synthetic. I think they can be helpful for growers and, helpful for making deadlines and yield improvements and quality improvements and things like that. I still kind of am in the school of thought of, these natural processes like breaking down organic inputs, we're probably missing something when we're applying just the pure compound. But further, science needs to prove that.
So if we're talking about getting these auxins and cytokines and gibberellins and putting them on, and that we're getting them from plants, it sounds awful lot like we're talking about biostimulants again. And perhaps there is an argument to be made that plant growth regulators and natural biostimulants are all in the bucket of plants that have got chemicals, hormones, and nutrition that are also used by the cannabis plant that we just want to wildcraft, or what's that term you used? Bioprospect, on our properties, and then just allow them to either degrade in their basic form or incubate them with, oxygen and then apply them to our plants. It sounds like it's the same system, just using different vocabulary.
Yeah, I would agree with you on that. I do think the PGR term fits nicely underneath the umbrella term that is biostimulants in my world, and maybe the world. But yeah, you think about how these compounds, mainly, the plant is naturally producing these things. When it needs roots, there's auxins being produced inside the plant to signal those meristem cells to turn into roots. But having furthered science in agriculture, we as growers can use these things to get effects that we want to. I know in a lot of the ornamental crop industry, things like anti-gibberellins are sprayed on flowers to keep them tight and not too leggy or stretched out from the pot. That way they make a nice compact bushy poinsettia or zinnia or something like that. So yeah, it's interesting to think about the use of PGRs in products that are going to be consumed by people rather than just enjoyed by aesthetically, like flowers and things like that. And I think I need to do more reading on the consumption of things that might be residual on flower if the plant wasn't able to use all them and or even just surfactants to get them to spread as well.
So when I have talked with other regenerative farmers about the naturally occurring PGRs that they use, I only have ever gotten one response. Either the farmers don't think of them in that way, and so they're just using degraded plant, compost teas or spun aerated teas. So they'll either say nothing, they don't use one intentionally, or they will say banana peels. And so is there any, this is kind of a nice pun, is there any low-hanging fruit that you recommend where we can go for the hormones that have the effect on the cannabis plant that we're looking for, so we don't have to go to synthetics? For example, if we want tighter flowers and a more compact overall plant, do we want to go to a banana peel, or are there things that we may have access to that you think might be better for us to go after?
Yeah, I think ultimately, I don't have any sort of materials off the top of my head on things like anti-gibberellins. I know seaweed is a good one for cytokinin. I think people have used, honeys for auxins, whether that's, I'm not sure if that's actually a root increaser or if it's just a good antimicrobial for, pathogens that might infect the plant before it can root. But yeah, I mean, I'm a huge composter, and I'm a big fan of vermicompost as well.
Yeah.
So I think, doing that stuff in the backyard when space allows, there's indoor systems that are really great too. I have a friend that did like a 3-bucket stack system where the worms were in the bottom, the new food waste went in the top, and when they ran out of food, they would kind of dig up and kind of make it a closed loop system. So there are ways to kind of get those organic materials broken down depending on the space you might have. The other kind of school of thought too is when I was a lot younger, I was always told, dig a hole 6 or 7 feet underground, bury a couple dead fish there, and that's where we'll plant our cannabis plants the following spring,
right,
in the fall. And it's been cool to kind of enter more of the sciencey realm of this kind of stuff and seeing like, oh, well, yeah, the fish breaks down into ammonium or some form of NH4 or 3, and the emulsions, and they provide nutrition from those breakdown products. So I just one thing that I love about the cannabis science industry is, or just cannabis science in general, is kind of seeing old school of thought growers with these really wise practices that we are just now starting to understand and can be measured scientifically with how does a fish break down in the soil and what types of plant inputs does it turn into.
Yeah, all of this old school cultivation magic, eventually science catches up with it and it's like, oh yeah, I guess, the old hippies were right in what they were doing.
Absolutely.
It's like, everything old is new again. So right on. Well, let's wrap up this first set here. We're going to take a short break and be right back. You are listening to Shaping Fire, and my guest today is microbiologist Michael DiLegge. Revenue is tight for cannabis businesses right now. There is no question about it. And when revenue tightens, businesses need to spend smarter on advertising. Instead of throwing obscene money at hanging your company's logo from the rafters at your next convention, perhaps consider placing commercials on Shaping Fire. And if you are a very small business, there is simply no better bang for your buck than commercials on Shaping Fire, especially if your products are for regeneratively minded cultivators. Advertising on this show allows us time to talk about your product, service, or brand in a way that really lets people know what sets your company apart from the others. Bold people who own companies know that getting into relationship with their customers is essential. That is what we offer. We will explain your service or product, what sets it apart as desirable, and help our audience get in contact with you. It's pretty simple, really. Advertising does not have to be all whiz-bang, smoke, and mirrors. Nowadays, I find that people prefer just to be spoken to calmly, accurately, and with good intentions. If you want to make your own commercial spot, you can do that too. With a commercial on Shaping Fire, you'll reach your customers in the privacy of their headphones right now, and will continue to reach new listeners as they explore the Shaping Fire back catalog of episodes again and again for years. A spot on Shaping Fire costs less than a postcard per person, and the Shaping Fire audience is full of smart cannabis enthusiasts, cultivators, and entrepreneurs who are always curious to learn. And right now in the new year, we are booking the few open spots we have for the rest of 2024. I'd love for you to be one of our new advertisers. Do yourself a solid and contact us today for rates on podcast and Instagram advertising. Email hotspot@shapingfire.com to find out more. As cannabis regulations become more demanding and consumers become more educated, it is increasingly important to avoid the use of chemical pesticides when cultivating cannabis. Beneficial insects have been used for decades by the greenhouse vegetable and ornamental plant industry, and today many cannabis cultivators are moving from sprays and chemicals to beneficial insects. Copert has the beneficial insects, mites and nematodes, microbials, sticky cards, and air distribution units you need to partner with nature to defend your garden. Whether you manage acres of canopy or have a simple grow tent in your home, Copert is ready to help answer your questions and help you transition away from chemical sprays towards clean and natural solutions. Since 1967, Copert has assisted growers in identifying pests and devising reliable solutions while providing healthy insects and mites that will protect your yield. Since the 1990s, Copert has been a leader in cannabis pest and disease control worldwide and have highly trained consultants to assist you in Canada and the United States from coast to coast. With their global network of grower support, Copert can help. Visit Copert.com, choose your country, and get detailed information. That's Copert, Koppert.com. For the most up-to-date cannabis-related biological control information, you can also check their Instagram at Copert Canada. You know getting away from pesticides is good for health and good for business, and Copert is ready to help. Visit Koppert.com today. There are so many seed banks nowadays that you really have options in who to choose. Not only that, if you pick the wrong seed bank, you could be in for a really sketchy ride. And that's only one of the reasons I recommend Gas Lamp Seeds to my friends and listeners who are looking for a seed bank. You probably already know Gas Lamp Seeds as Hembra Genetics. Hembra recently changed their name to Gas Lamp Seeds. Gas Lamp Seeds is not just another seed bank. Gas Lamp is a female-operated boutique cannabis genetics provider that only sells thoughtfully curated seeds from the top names in cannabis breeding. With over 60 breeders and over 1,000 strains to choose from, you will certainly find something you'll love. Gas Lamp Seeds has something for everyone, with over 650 feminized strains, 300 regular varieties, and over 200 autoflowers to choose from. Names you know you can trust, like Compound Genetics, Humboldt Seed Company, Night Owl, In-House, Fast Buds, Gnome Automatics, and Ethos. And we both know that there are other seed banks who will take your money but have no customer service. I invited Gaslamp to advertise on Shaping Fire after hearing so many good stories about them from my friends. They have A-plus customer service with lightning-fast response times. In most cases, Helene and Caitlin will get your order out the same day you place it. Most seed banks are simply not this organized or interested in getting your seeds to you so fast. But Gaslamp Seeds cares. You even get free seeds with every order. Helene and Caitlin get it. They have been in the cannabis growing scene for over a decade. Want some extra freebies? Use the code SHAPINGFIRE, all one word, at checkout, and they will give you an additional set of Gaslamp provided freebies. That's an extra $30 in free seeds. Buy seeds from good folks who will send you great seeds reliably every time. Visit gaslampseeds.com today. That's Gaslamp Seeds. Welcome back. You are listening to Shaping Fire. I am your host, Shango Los, and my guest today is microbiologist Michael DiLegge. So during the first set, we talked all about biostimulants and where to find them in nature, as well as the idea of plant growth regulators. And we kept on coming back to this idea that what we want to do to help our cannabis plants the most is to help the rhizosphere, to make sure that the nutrition and variety of microbes are incredibly varied, are indigenous so that they survive in our local natural environment, and that we can bioprospect them all over our own property and near us by just going to other plants that we already have locally. And not only does that save us money, but we know that these plants that are already surviving locally are already covered in indigenous microorganisms. So saves us money and is exactly the tool for the job to help you have your best grow ever. So here in set 2, we're going to talk about the care and tending of the microbe community that's in your rhizosphere, right? So these are all of the residents of the rhizosphere, the root zone, that make up the soil food web that is within the soil. As we all know, the soil food web also is above the soil, and it also takes place in water. But specifically, we're talking about this neighborhood of the roots of the cannabis plant. So when I was listening to Michael DiLegge on our friend Leighton Morrison's show, he, and I guess I'm talking to the audience, Michael, and to you kind of third person. You were talking on Leighton's show about your love of biochar, which we're big fans of on the show as well, and I really liked how you kind of anthropomorphized the microbes, because you were talking about how biochar has got all these ridges and places to live, and they've got these environments that the microbes can set up housing and where their nutrition grows, and how it just kind of makes for this perfect neighborhood. And I always like to think about my pots that way too, or the soil that I want to make a good neighborhood for my plants to grow in as well. So you and I clearly enjoy anthropomorphizing microbes, which I can support. Let's go ahead and start just kind of generally with this idea of creating neighborhoods that have got the kinds of attributes in the soil that these neighborhoods can build on. What is the nature of soil structure that makes good rhizosphere?
I think a good balance of sand, silt, and clay obviously is a great way to, that definition of soil makes a good structure for the microbes to colonize. But I think a really important thing that is often overlooked would be things that we call MAC, M-A-C, microbially forms of accessible carbon, or microbe accessible carbon. Your plants are, constantly depositing these things into the soil through root exudates, as well as, any organic matter and different types of breakdown processes in soil or other systems can provide these forms of MAC carbon sources, food sources for the microbes to be in there. Biochar being carbon-based is a really great form of this. Other things like fullerenes and different like, cellulose breakdown products are another great way to kind of feed microbes in the soil.
So is the primary, I'll say the first, not the only, but is the primary attribute of creating a good neighborhood making sure that there's lots of these macs around, so there's always high quality carbon for them to eat? Is that at the top of the list?
I would put that at the top of the list, just because if you apply something like a biological to a near sterile system, I'll never call a hydroponic system sterile, just because there's microbes everywhere, microbial ubiquity. But we do have, the notion of a lot of that stuff can be wasted if there's not food sources to feed the microbes. You hear about with your gut and gut health, prebiotics with probiotics, that's, feeding also the things that might be resident to your system, whether or not they're applied from a bottle or from indigenous microorganism hunting.
So what are sources of these macs that we can wildcraft, and how do we process them into it? I mean, the biochar, we can use as a soil amendment, and some people like to crush it up and top dress it into their water so it slowly works their way in. But give us some more idea of how to think about these macs and where we can bioprospect them into our cultivation.
Absolutely. Yeah. I mean, I think I'm going to do a callback to biostimulants, right? There's lots of macs in different tiers of biostimulant products or things that you can get or procure naturally with wildcrafting. And essentially, the plant is also feeding your microbes. So as long as your plants are maintained optimal health and nutrition, there's going to be some source of macs in the system from the root exudates too. But you will, plants also can reuptake root exudates. So there is a degree of plant microbe competition for these food sources. But going back to that kind of old wisdom cultivation, thinking, I hear a lot of people have really strong opinions about applying molasses as a input during flower. And I've always kind of understood that as we're feeding the soil microbes rather than we're, giving sugar to help increase flower synthesis or something like that.
Yeah, we've talked on this show many times how the adding of molasses into the pot, what you're doing is throwing your pot out of balance. What you want to do is, if you're going to use the molasses, is feed the microbes in your aerated compost brewer. Let them eat the molasses there and incubate them. So now you have a whole bunch of them, and then put that compost tea that's not hot anymore into your rhizosphere. Are those the steps that you're referring to?
Yeah. I think that is probably the best practice to do it for those who are unable to kind of have the lead time to brew a cup of compost tea and feed it sugar and whatnot. I think just a very diluted form and irrigation solution can also kind of benefit those microbes that might be in your roots.
Right on. I want to push you a little bit on specificity, because you said that there are various plants that we can wildcraft where we can get the max from. Just for examples to get us started, because we all know that we can Google it or ask GPT or something. But from you, will you give us 3 mac-rich plants that we might be able to wildcraft, just so people who just want to jump on it know where to start?
I think I'm always going to go back to compost and those breakdown processes. When I say, you ask for specific plants, I don't know that I have any of those off the top of my list, but I think, carbon and nitrogen sources essentially can help increase available max, whether or not they are being added to your system or the nutrients, the compost pile or something like that provided. But in terms of kind of going out and foraging up a bunch of plants and making a tea out of that, I would have to do some more research.
Right on. Your hesitation kind of suddenly recontextualized everything for me. Is it potentially the answer to the question I asked, that these macs are in everything because carbon is a building block? So long as I'm starting with a local, healthy, thriving plant, it's going to have max in it. So just use whatever I have a lot of around me, and use a variety of plants that are around me. And then if I want to up it, well, then go ahead and do some online research and find out the types of plants that are more rich in max. but I'm starting to get the idea, since we keep on coming back to compost, that the MACs are everywhere, and so just use whatever you have.
Yeah, I would agree with that. Thank you. And another kind of way to put it, I guess using whatever you have should be used with caution a little bit, as a lot of people know. If you look for broken down, dried out plant material, you want to make sure that didn't come from anything diseased. Obviously, you'd want that to be fully broken down in a composting system or a heat breakdown type of format before you add something that might be, degraded, a leaf loss due to a xanthomonas bacteria in the leaf or something like that, right? We don't want to reintroduce that to our system, but plant health is very important too when you're kind of wildcrafting.
Right on.
All that to say.
All right, so when we're talking about the care and tending of microbes, obviously we want to include a lot of these MACs so that there's a varied buffet all through the rhizosphere for all the varieties of residents to eat. So let's go, so long as you think we've covered that well enough, let's go beyond the nutrition for the microbes eating the carbon, and let's talk about some of the other attributes of soil structure that might also be beneficial to the rhizosphere. And again, I might be over anthropomorphizing here, but in my notes I put like shelter, reproduction space, non-carbon nutrition, like the idea of shelter may not actually exist in soil in the rhizosphere, but it seems like it should exist there because you have got more predator residents in the biosphere and more passive residents in the biosphere, and normally life forms need to take shelter when they reproduce, and so I'm very willing to be very wrong on this, right? But tell us a little bit about how the community interacts and if there are attributes of our soil and amendments that potentially provide more of this shelter and reproduction space and non-carbon nutrition.
Absolutely. Yeah. I mean, I think the best place for plants to overwinter in terms of seasonal changes would be in the roots and in the kind of skeletized plant bodies that are no longer actively growing. That's a great place for microbes to overwinter and be ready to be introduced to your plants when you're planting them in the springtime. And then I just do want to circle back a little bit on the MAC stuff and say one more thing. Sure. I want to go back to, I want to quote Dr. Elaine Ingham from the Soil Food Web and say, what do microbes love? Sugars, right? And that's why the cakes and cookies that are the root exudates are really kind of, being deposited by the plants in theory to colonize or recruit microbes to the root zone. The other thing that I wanted to mention was going back to that big open table analogy with empty seats. Another kind of enhancement of this theory is like, if that table is being catered and there is only, slabs of meat being put on that table, only carnivore type people are going to be sitting down there to consume that stuff, right? So we also want to provide a lot of diverse inputs of MACs, whether that is different types of compost tea or sugars like molasses or just broken down leaves and pine straw and stuff like that. But the important thing is by providing a lot of different forms of microbial accessible carbon, we're feeding lots of different functional microbes in the soil, right? So if there's if that table is being catered with vegetables, meat, and a little bit of both, we're going to have omnivores, vegetarians, vegans, and carnivores all sitting at that table. The same applies in my head, at least, to the plant roots, right? So if the tomato root is only exuding the same compounds tomato plants exude, then only a few types of microbes or functional microbes are going to be able to use those food sources. So the grower can kind of apply different forms of MACs, molasses, compost, stuff like that, or even companion planting to provide a lot of different food sources because the general thinking in microbiome science is health is very closely associated with high diversity in a microbiome. All
right. So with my next question, I'm going to set up a little a little example first about EM1. And, a lot of folks like to use EM1. And the idea is like, okay, you can buy EM1, which is a blend of microbes, and you can add it to your plants, and your plants will love it. And if you want to expand the bottle so your bottle lasts longer, there's usually instructions on the back of how you can do a simple aerated tea so that you can expand what's in your bottle, but eventually, if you just keep on brewing it, the microbes that are differently aggressive and competitive, the balance that is in the actual EM1 balance that they recommend, it'll eventually get out of balance, and so you have to go and get a new bottle of EM1 and start over with the actual balanced product. So using that as the example. If we are grabbing lots of different plants that are local to our area that are thriving, and we are either degrading them or extracting them into some sort of extract or aerated compost tea, is there any awareness we should have about the balance of those microbes and nutrition that we're actually putting into the pot? If we do want to have our rhizosphere life to be in balance, and we influence that balance by what we're adding from our compost pile as a tea, do we need to be worried about throwing off the balance of the residents of the rhizosphere because we are feeding out of balanced teas?
Yeah, actually, that is a major concern, and it's cool that a product like that has those types of instructions for kind of increase, bang for your buck. But yes, I think my best advice, and this is maybe my opinion, but based on some science, would be when you're making those types of products, try to use them up. Do your research and say you've found a lot of sources to say 48 hours is the ideal incubation or aeration time for a compost tea. Try to use that up pretty quickly after it's ready to go. And I think that is based on the thinking that all types of microbes, whether there's one thing in a bottle or 1,000 things from a sample of compost, have different growth and metabolism rates. So when things are grown together, the term is like co-cultured, the faster growers are always going to be able to have that advantage towards the other members of that biome because they are just genetically advantaged to eat faster and divide faster and break down faster, right? So you can see things like if you let a compost tea sit for 2 weeks, maybe if it's aerated as opposed to 1 week, I bet that microbiome completely shifts just based on the ability for those faster growers to become dominant. And I think, the diversity was probably the highest within the first 72 hours of brewing a compost tea. And that starts to probably taper off just based on the way microbe ecology kind of works in that system. There's only so much food around, right? And if the fastest eaters are, kind of throwing their elbows at the other ones and eating it faster, they're going to become dominant. And maybe those aren't the plant growth promoting microbes that you're in search for. Maybe they're just really good at eating those max.
Mm-hmm. I like that idea that as you're brewing it for 36 to 48 hours and you're pulling it out of the bucket right then, you have got so many aerated microbes and they've all just been getting all the aeration and nutrition that they could possibly want, which just means like everybody is fed and thriving and at the starting line, and that's when we want to give it to our plants. But the longer we get away from that starting line and let that compost tea sit for 1 day, 2 day, they're all already racing and competing and beating each other up and eating each other. And so by day 3 or 4, you're fully at survival of the fittest at that point, and you're not providing a nice, even, balanced array of nutrition and microbiology. You're actually providing them something imbalanced that's going to have some sort of effect on your soil environment. Does that sound about right?
Absolutely, yeah. I mean, and the way to say that is in microbiome sciences, we use a term called alpha diversity. So if I have a basket of fruit and there's 10 pieces of fruit in there and 9 of them are apples and 1 of them's an orange, I have really low alpha diversity. But if I have 10 pieces of fruit in a basket and one's an apple, one's a grape, one's a banana, blueberry, everything is a different thing. The parts of a whole are all different. That is really high alpha diversity. So the longer something like a compost tea might sit, it's likely that you be lowering your alpha diversity, where those fast growers or good eaters are going to become dominant.
Right on. That is super interesting. And because I want to look this up after the show, are you saying alpha, ALPHA?
That is correct, yes.
All right, cool. That's a very interesting idea. That is new to me.
On compost teas as well, I collaborated on something when I was in school where a colleague was making vermicompost tea from worm castings and finished compost. And this person was able to grow it up. I think they did a 48-hour aeration, and then they boiled half of it, and then they applied the other half untreated. And they actually found that, I think it was a basil host or a tomato host, those plants benefited more from the boiled compost tea. So this is not me suggesting that everybody should be boiling their compost tea, but maybe on their aeration time, they were able to, increase the populations of things that might not directly benefit plants and actually compete for plants for those beneficial parts that get released when we make a compost tea. So it was kind of interesting to think about, boiling or trying to get the microbes out of a compost tea, the ones that can't survive boiling, actually made the plants do better, presumably from a reduced amount of microbe competition for the same things the plant wants that kind of get released from the aeration process in compost tea.
Wow, man. Boiling compost, you just kind of blew my mind there. I'm going to have to think a lot about that. But what I think about it immediately is that if we've talked constantly throughout this episode so far about the biodiversity of the teas and, of the carbons and of the plant sources that we continually pour into our plants, that if I'm going to make aerated compost teas and I'm also going to do cold extracted teas, like I don't see any reason why having a big pot of boiled plants that I then let cool and then apply to the plants, because I'm going to get a whole different cast of characters in that, and all I'm doing is increasing my diversity by using all 3 different types of teas.
I would agree with that. Yeah, definitely. The non-boiled compost tea in the study I'm referring to is kind of a balance of both probiotic or just bacterial members and prebiotics, whereas likely after it was boiled, it wasn't measured chemically, but that was probably just more, I'm going to use the term prebiotic, but just kind of the metabolites that are released from the compost tea brewing process, ready for the plant to slurp right up rather than kind of have to share with the microbes.
Right on. All right. So before we move on to our next topic, I want to hit something one more time because, I've heard you kind of allude to the fact that one of the good things that biochar does in the soil structure is to, not only provide, the carbon for food, but also it's like a unique housing structure, if you will, because it's got all these nooks and crevices. Is there anything, and the answer may be no, but I'm going to try to squeeze you on this anyway. Is there anything else that we should consider adding as an amendment to our soil that is less about nutrition and having it break down to provide, foods, which is what most of the stuff we do is for, but instead is something that we are going to amend the soil because it actually provides a physical mechanism that is good for the microbe life?
Yeah, for that, I think, the biochars are a really good approach to that. And I think I'll always recommend, if people aren't already doing this, to get a starting soil test before you start applying your inoculants or inputs to that as well. But I think in terms of kind of like housing the microbes or giving them like a nice place to live, again, I'm biased towards the soil systems as opposed to like a hydroponic system. But when we think about no-till, not only is that really kind of maintaining soil, carbon that's stored from those plants, but that's also keeping those houses for the microbes dormant in the plant roots, ready to be introduced to the fresh roots that would be kind of germinating in the same soils. So like me personally, I have one of my experimental, garden beds in my backyard that I've never tilled it for, and I'll plant seeds or directly transplant things there. And it seems to be, speculative that those plants get a little bit of a boost because I do apply heavy compost and things like that. There's a lot of factors we're not considering, but I think a big thing is, those microbes have overwintering structures, and they were evolved with the plant to survive through these cold temperatures and hot temperatures. I think people should maybe experiment more with no-till or, companion planting and things along those lines.
Yeah, that certainly makes sense regarding our experience with no-till. Every year, the soil just gets more mature and it lasts better through the winter, and the following year's plants are thriving. It's always hardest that first year when people are using immature bagged soil without a lot of mycelium, and they get kind of mediocre plants. And then the longer they go no-till, the more beefy the plants get.
Yeah, it's like a bettering. I think there's probably a limit to it. Year 3, year 4, something like that. Everybody's system's different, but yeah, the more organic material and different types of plant materials and their root exudates, stuff like that, you can get in that system. You're adding that diversity, which is really correlated with plant and microbe health.
Rotating and amending, no-till soil every, let's say 4 years, that is such a massive improvement than the idea of, kind of old school where every year you start with new, fresh, clean soil that doesn't have any pathogens in it. And while we want to avoid the pathogens, but the best way to actually keep pathogens at bay is to have a vigorous soil that's already aged. And I always feel bad for people who are very much set on the new season, new soil tip, because it just makes things hard every year instead of year 3 and 4 being like easy.
Yeah, to me, that kind of feels like a scorched earth type of approach. And I like that you mentioned, the diversity in what you just said. We've seen things like Fusarium species highly present, 10 to the 6th, 10 to the 8th cells per gram of soil in a plant rhizosphere sample. But it's, very far along into flower maturation, and there's no signs or symptoms of the Fusarium attacking the plant. So it's like the Fusarium is taking one seat at the table with 100 seats, and the other 99 seats are plant benefiting things or soil benefiting things. So, pathogens can be present without becoming virulent, and it all comes back to soil microbial diversity.
Out-competing Fusarium is the only potential solution that I see that there is, other than just dumping the soil. We've talked about Fusarium. I've had Fusarium specialists on the program, and usually their answer is like throw the soil away, and I get that. But it's interesting to realize that as a preventative, since Fusarium's everywhere, it's not like it's not going to be in your soil. Just make sure you keep on, adding microbes, incubating microbes, adding indigenous microbes, and Fusarium will never get a good foothold.
Yeah, I mean, never is a strong word, but I think
fair enough.
Differently from, adding all the microbes and really intentionally applying, diverse forms of microbes, whether they're from, your wildcrafting or input somebody could buy. I think the other thing is, soil health practices really is where a lot of this soil microbial diversity preservation can start. So those things like the prebiotics and the organic inputs and companion planting, crop rotation, no-till, soil health, I think maybe even I would put higher on the list than microbial accessible carbon from the previous question, but they're related, right?
Yeah. All right, cool. So the last topic I want to hit during the set 2 here is, so far this set we've been talking about the things that we can do to make the living environment for the microbe life and in the rhizosphere as diverse and beneficial as possible, and why that's kind of everything, right? We keep on coming back to the same answer, and it's because the answer is kind of the only thing that we need to be doing as regenerative farmers is adding diversity into the soil, and let nature do the rest. So that's great. I also am very attracted to the idea that the plant is also trying to help itself as well by putting out, exudates that encourage particular microbes or different residents in the rhizosphere, where they're kind of curating who they are attracting to them. And I've heard you talk before about recruitment, that cannabis plants will recruit bacteria that they find beneficial. And I'm unfamiliar with this idea, but just the idea of it is fascinating. So I don't know the right question to ask you on it. So I'm just going to kind of hand you the mic and say, will you tell us about how cannabis plants recruit beneficial bacteria itself, kind of like intentionally?
Yeah, absolutely. Thank you. I think, there's a lot of literature from the past 5 or 6 years that show photoperiod sensitive plants, or plants that, have major changes in development, also change their root exudate profiles. So I think it was first, learned about in a Arabidopsis wild mustard model, a plant that has a rosette stage, and then it bolts, and then it makes flowers, and those flowers make seeds. At each one of those stages, it was seen that the root exudate profiles change. The early growth and development, there was just a ton of sugar, and later in the flowering and maturation, there was a lot of organic acids and things that are can be harsh to certain microbes, whereas sugar is like a really broad food source. So the kind of thinking from the authors of that paper were, and I'll get to cannabis too, I promise, were about the developmental needs of the plant are changing. More nitrogen in the beginning, more phosphorus and potassium later, and those micronutrients trickled throughout. And we know that the microbes help plants uptake these things. So, let's wonder if the plant is actually, I'm going to use the term intentionally, signaling for different things as it goes on through growth and development. So, being a cannabis enthusiast, seeing that study, I was like, wow, what's the most popular photoperiod sensitive plant that we use? Hemp and cannabis, right? Mm-hmm. So it was, I'm not sure if root exudate work has been done, but we've done some work on the vegetative versus the transition. So, maybe a couple weeks after flower onset, after the solstice, and the days get short in an outdoor system, as well as the harvest, functional microbiome of cannabis. And we see that, regardless of the, specific species of cannabis or strain, whether it's hemp or cannabis, 0.3% THC producers or higher, the same functions, or at least the same bacterial genera, are occurring in many different types of soil environments. We're using like the USDA climatic zones for that. But basically, cannabis being as nutritionally needy compared to some other plants, we see really strong associations with the bacteria based on development. So when we use the term like recruit, it's kind of like the cannabis plant is sending these signals into the rhizosphere to likely get some type of rhizospheric function by the bacterial or fungal or just anybody in the soil microbiome. And these needs are pretty standard from, the genus or the cannabis sativa plants, whether they are classified as hemp or marijuana, depending on the amount of metabolite it produces, right? All that to say is plants really want microbes. And if we can kind of provide microbes that the plant can't search for in indoor systems, or in really, low alpha diversity, 10-year monocultured cornfield soils where there's like 5 different types of microbes, I'm exaggerating, then we can kind of benefit the plant in a way that it would in its natural environment, in the wild type relatives and the progenitors might have, kind of evolved to do.
So it sounds like, if I'm understanding you correctly, the main point is that over the life cycle of the plant, the dominant bacteria is in the rhizosphere changes, and we know the mechanism, or at least we're pretty sure that the mechanism of why it changes is because the plant wants different types of bacteria, and so the plant is putting out different types of exudates to encourage different types of bacteria, and so the best thing that we can do is to, let the plant do its job, and then B, do our part by offering varieties of other nutritions and probiotic bacteria and such that the plant can't produce and it needs to get its environment, so that between the partnership between us and the plant, the roots have just got everything it could possibly desire to express itself most fully.
Yes, that is a perfect distillation. Thank you. And I think, most importantly, I'm going to sound like a broken record, but soil health, right? If the soil health is maintained, then we are going to assume, and we've proven, like the science has proven, there's lots of alpha diversity, microbial diversity in a very healthy soil. So whether or not the microbe that's present during the vegetative growth period of your cannabis plant's life cycle is recruited by the plant, during that time, if you maintain that soil health, it's likely there will still be those microbes in low enough cell numbers. And then when the plant changes its root exudation compounds, presumably to attract microbes to help it meet its nutritional needs based on development, then, soil health maintains microbial diversity. Those bugs are hanging around when your plant is flowering and in need of those types of functions.
Hell yeah, that's actually really exciting. I like that summary a lot. It's like it's very rare in discussing soil health and soil maximalism, if you will, to actually have that clear of a recipe for what we need to do this season. Thank God. So
yeah, right.
Right on. Well, good. Well, thank you for that, Michael. Let's go ahead and take our second commercial and be right back. You're listening to Shaping Fire, and my guest today is microbiologist Michael DiLegge. And remember, without these advertisers, Shaping Fire wouldn't happen. So please support them and let them know that you heard of them on Shaping Fire. You've heard me talk about the award-winning cannabis seeds that come from the analytical breeding program of Seth and Eric Crawford, who founded Oregon CBD Seeds. In fact, Seth was a guest on Shaping Fire in 2020 to talk about triploid genetics. Seth and Eric are now releasing high THC seeds for home growers and farms as Grow the Revolution Seeds at gtrseeds.com. Their high THC seeds are extraordinary in that they will start to flower at 16 and a half hours of daylight instead of the typical 14 and a half hours of daylight. That means in most regions, your plants will start to flower outdoors in the middle of July instead of the middle of August, which means these photoperiod plants finish in September and not October, totally upending the photoperiod seed market. Seth and Eric took their prized early flowering CBG line and bred it to some of the most desired verified genetics out there, including Sour Diesel, Triangle Kush, Wedding Cake, Chemdog, Skittles, and others. These crosses all express powerful photoperiod terpene profiles and THC, giving you a great high. GTR Seeds has a new THCV line with plants like Double Durban and Gigantor that boast 1-to-1 THC to THCV, and people want that THCV. GTR seeds are very consistent, true-growing, inbred F1s from stabilized inbred parent lines. These seeds are nearly homogeneous, and the plants should all grow the same. There is only one phenotype in every pack, available as diploids and triploids. Seth and Eric's company is still family-owned, patient and employee-centric, and partially powered by their 2 acres of solar panels. Everyone can purchase these seeds and the entire catalog of Oregon CBD seeds at gtrseeds.com. Go to gtrseeds.com today and choose something revolutionary for your next indoor or outdoor run. Fish Poop Brand Fertilizer is an all-natural fish poop concentrate with nothing added. Real fish poop is extraordinarily complex. Not only are you adding the nitrogen, phosphorus, and potassium your plants need to build mass, transport nutrients, and enhance flavor, but fish waste is also packed with biological activity and micronutrients. When you add fish poop to your irrigation water, you are adding life force, probiotics, and active microbes. These microorganisms include a wealth of various bacteria and protozoa which further enhance nutrient availability for the plants. Because plants are limited by the absence of any essential micronutrient, these trace nutrients are the difference between having a decent garden and having a garden that makes you feel really proud of your efforts. Fish poop is a naturally complete solution that fills in the cracks in your fertilizer program to ensure you offer your garden a broad base of nutrients. Not all fish poop is created equally. Most products with added fish waste don't reveal their sources or lab results. Fish Poop brand Fish Poop, however, generates their own fish waste as a byproduct of their organic aquaponics cannabis farm where they raise ornamental koi and tilapia. You are even invited to tour their farm in person or on their YouTube channel to look for yourself. This sort of transparency is wildly rare in the fertilizer market. The folks behind Fish Poop are also lifelong medical cannabis producers who have deep connections in the community, donate more product than they sell, and support cannabis prisoner, veteran, and patient collectives and charities. To get your bottle of pure fish poop, go to fishpoop.com. And to see their entire line of cannabis products, go to ounceofhope.com. That's Fish Poop brand Fish Poop. One of the reasons why no-till cannabis growing is so valued by farmers is because the mycelium networks in the soil remain established from year to year. And we know these fungal networks are essential because they are the nutrient superhighways that extend far and wide in the substrate to feed your plants. The trouble with growing in new living soils or blended cocoa substrates is that it takes most of the plant's life just to create these mycelium highways. Dynamyco endomycorrhizal fungi inoculant reduces that time and gets your plant eating a wider array of nutrients faster. And it's 3 times the concentration of the other popular brand in the US at 900 propagules per gram of 2 fungal species selected specifically for cannabis cultivation. Dynamyco is the result of 30 years of research and trials at the Volcani Agriculture Research Institute in Israel. It has also been vigorously trialed by cannabis and food growers across the U.S. Dynamyco is now available at grow shops and on Amazon in the United States. I love using Dynamyco to both speed up the growth of the mycelium networks in the soil, but also as a biostimulant to make clone cuttings more virile. You can see side-by-sides showing the comparative growth on their Instagram at Dynamyco. If you demand reliable growing results and appreciate the importance of an active root zone in creating a thriving plant, I encourage you to check out dynamyco.com and use the store locator to find out where you can get yours. That's dynomyco.com. Shaping Fire listeners can get 10% off any size of Dynamyco on dynamyco.com by using the discount code SHAPINGFIRE, one word, no caps. Whether you are starting with new beds or pots, or if you want to add some zing to tired soil, choose Dynamyco to maximize your plant's potential. Dynamyco endomycorrhizal inoculant. Welcome back. You are listening to Shaping Fire. I am your host, Shango Los, and my guest today is microbiologist Michael DiLegge. So here we are at the big finish. During the first set, we talked all about biostimulants and plant growth regulators and what we wanted to do as regenerative cultivators to make sure that we have the healthiest, most biodiverse soil so the plant can do its job of growing in a healthy way that resists pests and gives us awesome yields and terpenes. During the second set, we talked a bit more about the care and tending of microbe life specifically, and not only how we can add microbes and biodiversity like we were talking in the first set, but also how we can help them thrive and keep them well fed with carbon. And here on the third set, we're just going to hit on 2 more kind of miscellaneous biodiversity topics that didn't fit in the other 2 sections, but I think are really important and things that we have talked about prior on the program. So the first of the 2 is choosing companion plants to encourage biodiversity. So Michael, we've talked quite a bit today about plant exudates and how plants push out the exudates from the root structure and attract different microbial life and life forms in the rhizosphere that it wants to use as different flavors of tools. And I understand that like the plants that are planted near our cannabis plants can influence the rhizosphere of our cannabis plant because it is also calling forth different types of nutrition and mycelium to its root systems as well. And I understand, partly what you study is how these systems may interact and how to choose companion plants that will be a good neighbor for whatever the target plant that we're looking to grow. In our case, we're talking about cannabis. And so will you kind of talk us through how to think about choosing our companion plants and in order to encourage the most biodiversity in our soil?
Yeah, absolutely. And I think the reason that this isn't more, widespread or adopted rather is because of the scalability of this. But I would love to see it, on hundreds of acres. I think two really cool examples that were new to me for kind of companion planting for cannabis would be I had some friends do the Three Sisters, but replace the corn with the cannabis plant as opposed to corn. And for those who don't know, the Three Sisters is corn, beans, and squash. The corn serves as a trellis for the beans to grow up. The beans have a nitrogen-fixing symbiotic bacterium called rhizobium that live in their roots, and that nitrogen can get leached out of their roots and be available for both the corn and the squash. And then the squash, with their large leaves at the base of the plant, they provide shade, so soil stays kind of cooler temperatures and doesn't get baked from the sun. So in this case, they replaced the corn with the hemp plants and the cannabis plants to serve as that trellis for the beans to climb up. And both, the squash and the, the hemp was able to benefit from the beans being present due to that rhizobium bacterium being there. And also, certain soil temperatures, if they get too hot, that can actually be detrimental to microbial populations. So having that cooling effect from the squash leaves, depending on your growing environment, Colorado, we have 320 days of direct sunshine that can help cool it down to keep those soil temperatures happy for lots of different things to keep growing rather than just, the ones that are tolerant to warm soil.
Before you move past, the Three Sisters, I have got to ask, are you serious? We want to have beans climbing up our cannabis plants? Like, that sounds awful. Like, does that work? I imagine that the bean plants get all into the flowers and how does that work?
I think it might require a little bit more maintenance from the cultivator than most people can do in a production system. But yeah, so they would kind of climb up and on the fan leaves and things along those lines.
Do the
beans are also
do the beans stay closer to the stalk? Because like normally the flowers are kind of far away from the stalk. So are the beans perhaps going to just like grow up the center of the plant and not grow out to the flowers, or will the beans eventually grow out the branch to the flowers?
The ones I saw in the system were more so like the tips of the bean vines were kind of hanging out in the empty space between fan leaves in the middle parts of the plant. They didn't quite make it up to that top cola. This is a one example, right? So another approach to this, though, because I do hear what you're saying with, beans could effectively act like a bindweed and pull plants down that. So I think a bush bean or a different type of legume like a cowpea might be a better type of nitrogen fixer to get that, diverse root exudate profile, diverse microbiome from the root exudates, and the nitrogen component too from the legume plants.
And so those different types, those bush beans, and the reason why those are better is because you would plant those at the foot of the cannabis plant, and so their rhizospheres could talk to each other, but the plant is not a climbing bean, and so you'll just have like bean shrubbery at the bottom of your cannabis plant, which is fine. It just won't climb the plant. Is that where that goes?
Yes, absolutely. And that rhizosphere becomes representative of at least, 2 to 3 different types of plant species. With your vining squash, your fruits should be okay because they'll be far away from the rest of the beans that are kind of bushing at the base. But if you have all those roots kind of in the same root ball, as long as there's adequate nutrition and organic material, that will be like a very healthy, diverse microbiome as a factor of just having 3 different hosts.
Yeah, I like that a lot. And we've talked on the show before, too, that people who grow in berms that are heavy built up with compost and, grass clippings and other agricultural clippings, it's very popular to grow potatoes in that, too. So, you've got your potatoes and your beans and your squash kind of trailing off down the line of plants. I've always loved the idea of growing food crops with the cannabis so that you can feed yourself as well as smoke and save your soul, if you will.
Absolutely, yeah.
Okay, so now that I've pushed you on that, what other strategies or plans do you have in mind for creating a diverse rhizosphere?
Well, this was something that was kind of new to me that I read about. I didn't get to see this one in action, but it was a farm that had been co-planting a plant with a mushroom. So they were kind of using myceliated beds of a wine cap mushroom to get both harvest of wine cap fungi, edible mushrooms, at the base of the soil, and the hemp was able to grow and flower at the top. So it was kind of getting both that fungal microbiome benefit as well as the plant roots there too. So not directly plant to plant signaling like we're seeing with like the Three Sisters we just discussed. But something along the lines of, trying to have more bang for your buck in terms of square footage for harvesting and things like that.
And the wine cap mushroom is one of my favorite eating mushrooms. They're absolutely delicious. I tried to do that in my indoor, and it didn't work out because the humidity for mushrooms seems to be a little higher than we want it to be in an indoor setting. It seems to work well outdoors, so long as you've got good plant coverage, because normally we want our plants in direct sunlight, right? But the mushrooms don't want to be in the direct sunlight. So you've got to make sure that you're growing plants that are going to be big enough to create that shadow underneath that allows the wine caps to exist.
Yeah, maintain that humid microclimate underneath the plant canopy that is super ideal for mushrooms.
And the squash will help with microbes. That too, that you're talking about.
Yeah, absolutely.
Those big leaves. Yeah. All right. Do you have any more on that before I move on?
I think just the companion planting is really, really excellent, and I think there is a go back to the literature and look out. If you have deer eating your hemp, marigolds can be deterrents for deer, right? I think there's a different plant for a different need, and just always going back to the more diverse roots and hosts in the soil would increase your soil health and microbial diversity there.
So I actually was planning on doing big pots of marigolds next to the plants for the first time this year, because we do have a lot of deer pressure, and luckily I got some deer fencing this year, so hopefully that'll help too. But is it as effective that if the marigolds are not in the pot with the cannabis or in the ground next to it, but like are beside it, that the smell of the marigolds is enough to keep the deer off? Or will the deer just come over and browse the weed and just ignore the marigold?
I'll be fully honest with you. When I tried to use marigolds as a deterrent crop in Maryland, I watched a deer chew one down before. So that's kind of like
your mileage may vary,
right? Yeah, kind of like different strains of cannabis might have different terpenoid or cannabinoid profiles. Different species of marigolds, they have the multicolored ones, have different type of phytochemical profiles, or those volatile organic compounds that smell icky to deer. So I don't know a variety off the top of my head, but I imagine that it's very species specific.
Right on. Well, thank you. I appreciate your honesty in that. And if we're really going to go for it, these bioregions have got different deer, right? So different deer could be accustomed to browsing different types of plants, whereas Maryland deer don't like marigolds, but do like marigolds. They do like marigolds, and maybe my West Coast deer are more snooty and they don't want marigolds,
right,
or something like that. So, all right. But before we leave the companion planting topic, I just wanted to ask: are there any plants that are obviously poor neighbors? The name for it is escaping me at the moment, but there are often plants that you don't want to plant near other plants because they might poison each other or be toxic to the other person's or the other plant's rhizosphere. Are you aware of any of those?
Yeah, there is one that's jumping off the top of my head, although I know there's, a lot of examples. Black walnut trees, for example, not specifically related or cultivated for a regular harvest like we're talking about with cannabis or vegetables. But those guys have a root exudate that is under the nature of allelopathy. So they are actually able to shoot a compound into the soil that makes it pretty inhospitable for anything but its offspring to grow. And to counter that, there's also plants, I think certain trees as well, that don't want its own offspring to compete with the mom plant for the resources there. So they'll send signals into the soil that make it inhospitable for the plants of the same species to grow, to try to preserve its own nutrients that might be in the soil.
Right on. That's good to say. And I'm going to throw in my two, even though some people like these. I always make sure that any plants that I consider for a companion plant does not give off floating seeds like dandelions. I personally really love pink dandelions, and I encourage them in my yard actually because they look really trippy. And so, but before they go to seed, I've got a different seed crop at a different property, but like I have to make sure, I grab those because I don't want all those like little fuzzy puffballs in my resinous flowers, right? That is definitely a party foul. And so any plant that gives off, fluffy seeds that are going to be wind distributed. You want to be careful of those. And then even though a lot of people like red clover, I personally caution people from the red clover because the red flower shoots up. And if you don't grow monster plants, if you're growing plants that are only, 3 feet and not 10 feet, 3 feet, the clover could totally grow up into and start interacting with your resin, and we don't want that either. So the last question here is one that I talk about sometimes on Shaping Fire, but mostly the discussion has happened on the Shaping Fire YouTube channel, where I posted a video with a microbiologist that was a specialist in plant microbe interaction, talking about how the 24-hour light strategy is bad for cannabis. And I fully believe this to be the case because, photosynthetic plants have evolved processes that happen during the dark cycle. So if the plant is naturally growing outdoors, it gets a day cycle, gets a night cycle. During the night cycle, the photosynthesizing stops and other processes happen. And that's how you keep a strong, rambunctious plant. But we know from experiments, indoor plants, when you're supplementing with light, that if you grow the plant at 24 hours, it's like giving the plant cocaine or something, like where the plants just go. It constantly photosynthesize, constantly process these sugars, go, and so it will make your seed germinate faster. It will make the young plant move faster. It will bring it into becoming, a ready to flower teenage plant faster. And then in the flowering, some people find that they reach maturity at the flower faster to such a degree that they can, knock a week off of flowering sometimes. And they're all like, listen, if I do that with every cycle my whole year, I can actually squeeze in another cycle a year, and that's real money. And so because of that, the case for a 24-hour light cycle is made. And I take every opportunity I can to try to teach that the dark cycle is necessary for plant health. And you, Michael, you're the front line of this debate as a microbiologist. You know these cycles intimately, and you know what happens in the dark. And so I'm going to kind of throw myself up. Hopefully I'm not wrong in your eyes, but I'd like to hear you speak to this idea of the 24-hour light strategy and how that might impact the plant.
Yeah, absolutely. I think, that's got to be one of the most nutritionally expensive types of cultivation practices, I would think, that could be done. I wonder what types of, nutrient usage rates people are going through if they're doing this practice with 24-hour light cycles, because I would imagine that with 24 hours of daylight without any types of resting period, that the plant is going to be constantly requiring extremely high concentrations of nitrogen, phosphorus, and potassium. Essential processes like the Krebs cycle happen at nighttime. Plants also do these things called like guttation, where there might be air bubbles in the plant vascular tissue, which can be detrimental. But at nighttime, they can push those out. They also close their stomata and don't evaporate the water out of their bodies as quickly. So I think primarily the benefit to get another cycle might be missed by the amount of nutrients somebody might need from trying to maintain the nutritional needs of the cannabis plant by blasting them with 24 hours of light, right? If you're going to be, basically giving your plants cocaine and keeping them at high energy, high growth, optimal, fast growth rates and developments, there's going to be a lot of energy spent for that. And in order for them to kind of account for that deficit, there needs to be tons and tons of available nutrient inputs there. And I don't know that one other cycle might be beneficial enough to have to add that much more fertigation.
In my experience, the people who are defending the 24-hour light cycle are usually people that are using, 2-part bottled salt fertilizers. And so they're kind of like force feeding the plants nutrition anyway. And, it's pretty rare to find a bottled nutrients grower who isn't overfeeding anyway, because if some is good, more is better. Right. And so that's actually the answer that I get online when discussing this with these cultivators are at that. Well, if it just makes the plant more hungry for more nutrition, I'll just pour more in. And I'm like, that's not really how it works. But for folks who aren't familiar with it, you mentioned that the Krebs cycle happens in the dark period. Will you explain what that is?
Well, the plant starts to respire, so it stops using the light or the photons from the sunlight or your LEDs or any types of lighting you have as kind of like the building blocks to metabolism, and it starts to respire. Basically, it's getting its energy from off-gassing things that might be in there, as opposed to using the light energy to kind of make more chloroplast and things along those lines.
So when I'm talking about this with folks who are very, very into the 24-hour light strategy, they come back to me and they're like, well, you're telling me I can't do this, but I do this all the time, and I get plants and I smoke them and I get high and it's great, and to the best of my scientific knowledge, I say to them, yeah, but you're getting a plant that isn't the best plant you could have grown. If you need fast, cheap, styrofoam tasting flour quickly, okay, that might be an appropriate strategy to consider. But since most of us are looking for a thriving plant, which also provides high quality terpenes that just like engage us and give us a better high, and also creates pest resistance, it seems to me that the 24-hour light cycle will slowly but surely degrade a cannabis plant throughout its entire life cycle. What are your thoughts about that? Like, is the life of the cannabis plant so short where over that life cycle it doesn't matter if we starve the soil?
I kind of am thinking that it does matter. The cannabis plant life cycle is a lot longer to maturation and harvest than a lot of other different crops that are kind of following the school of thought. And I think that, speculatively, there's a lot of things that happen that are beneficial to the plant vascular system during the rest period, pushing those air bubbles out, closing those stomata, and things along those lines that I would imagine play a role in the synthesis of cannabinoids and terpenoids and other secondary metabolites. So I think, it would be interesting to do a side-by-side with the same kind of cultivar or strain of cannabis, in a 24-hour cycle versus an 18-6 followed by a 12-12. And then the other thing that I wanted to mention, you were talking about kind of the indoor growers and having those, 2-to-1 salts readily available and probably slightly over-applied. It would be cool to get to a point to where we can kind of start to supplement those conventional salt-based fertilizers with more organic things and maybe even get to the point, and this is from Dr. James White, the rhizophagy scientist, using those salts that are easily slurped up and immediately used in building blocks of the plant body, kind of as like corrective treatments. You notice a deficiency, you apply a salt, and then you start to kind of balance both your salt and your organic base of nutrition inputs. Not only will that benefit the plant, but also same thing with the microbes. There's been studies showing the same exact strain of cannabis growing in living soil versus growing in hydroponics with biologicals, and the terpene and cannabinoid profiles are statistically different from one another. And I think a lot of that has to do with the terroir or the terroir of the cannabis plant.
Yeah, the idea of supplementing with salts on its face, I'm prejudiced against, but as a citizen scientist, I want to learn more. You know what I mean? So
yeah, I understand. Yeah.
So anyway, well, thank you for diving into that topic for me. It's clearly something that I am passionate about, and I get argued with sometimes. And so anytime when I get a microbiologist like you willing to talk to me about it, I always bang that drum. So thank you.
Yeah, absolutely. I mean, I guess more on the microbe side, I think just like the soil temperatures do cool, or the root zone rhizosphere temperatures will also cool in the dark. So kind of factoring in the microbiome, that's just allowing more environmental changes to maintain different diversity and things like that with the darkness period, because a grow room will cool off 10 degrees when the lights are off and things like that.
Yeah, right on. Yeah, and I'm sure there's different cycles that need a cooler temperature to happen.
Yeah, but I'm interested in kind of learning more about the effects in the microbiome, with this 24-hour versus getting that rest period that plants, were evolved to have.
Yeah, so awesome. Well, Michael, we are here at the end. Thank you so much for sharing your expertise and experience and time and good cheer with us. I've really enjoyed our discussion, and I didn't expect so many times for us to come back to the answer being diverse microbe teas, but really, the more I get into regenerative growing and really growing in a way that's responds to nature, the more I realize that the farmer's only job is to create rich, nutritive, biologically diverse soil, and so you're always going to come back to tea. So thank you very much for being here and for sharing what you know.
Absolutely. Thank you so much for having me, Shango. It was a great conversation, and I'm really grateful to be able to kind of just, talk about this stuff with somebody else.
Excellent. Yeah, me too. It's nice to find another nerd for this stuff. So thank you.
Totally. Yeah.
Right on. So if you want to reach out to Michael or know more of what he's up to, there are two places to do that. You can follow Michael DiLegge, that's DILEGGE, on LinkedIn, where he posts what he's up to in his latest research. And also the company where he is director of microbiology is Impello Biosciences. And you can find out what they're up to at Impello, IMPELLO bio.com. You can find more episodes of the Shaping Fire podcast and subscribe to the show at shapingfire.com and wherever you get your podcasts. If you enjoyed the show, we'd really appreciate it if you would leave a positive review of the podcast wherever you download. Your review will help others find the show so they can enjoy it too. On the Shaping Fire website, you can also subscribe to the newsletter for insights into the latest cannabis news, exclusive videos, and giveaways. On the Shaping Fire website, you will also find transcripts of today's podcast as well. Be sure to follow on Instagram for all original content not found on the podcast. That's at Shaping Fire and at Shango Los on Instagram. Be sure to check out the Shaping Fire YouTube channel for exclusive interviews, farm tours, and cannabis lectures. Does your company want to reach our national audience of cannabis enthusiasts? Email hotspot@shapingfire.com to find out how. Thanks for listening to Shaping Fire. I've been your host, Shango Los.